Well, thank you so much. I'm going to to speak on on behalf of the on behalf of the people of my laboratory about the anti-ulcer properties of Anoda cristata, which is a Mexican edible quelite. So the term quelite derives from the Nahuatl, which is an autochthonous language in Mexico, and the word quelite means as that tender comestible herb. So these plants known as quelites comprise more than 500 species, and they have been consumed in Mexico since pre-Hispanic times until now. And in fact, nowadays they are exported and they are really appreciated by other countries in North America, like the United States, but also from Central and South America. So, considering its definition, the quelites include edible green, young, and tender annual plants, and that usually are not cultivated on purpose, as they tend to grow up around other crops. For example, like the crop of maize that we can see in the photograph. And the way of consumption may vary depending on the population or the geographical location, but they are often eaten raw or sometimes slightly cooked in boiling water. And in fact, some of them can be used for the preparation of beverages, as we can see here in the image, but the way of consumption also depends on the species. Unfortunately, quelites are not appreciated by consumers nor by farmers, and they are considered now as neglected and underutilized species because their consumption has been displaced by other major crops such as maize, rice, and wheat, among others. And well, despite their importance in local traditions and despite their nutritional value, in general, there is a lack of appreciation of these species, mainly due to social factors and cultural factors. And altogether, these provoke that they are not appreciated by the consumers. By the consumers, and they are not usually included in the research or agricultural agendas. So they, unfortunately, they have been wrongly called as the food of the poor. And again, without considering their importance or their nutritional value. So with the aim of promoting the research, the rescue and consumption of quelites, and trying to reintegrate them in the markets and in the daily diet of the Mexican population, it was developed a multidisciplinary project, which included the study of quelites from different points of view. And our participation mainly focused in the area of medicine. And our objective was to determine if these quelites, besides the nutritional value, could have the potential of being used for the treatment of pathologies related to the infection of Helicobacter pylori. The reason for studying quelites from this approach was because they are not only used as food, and as a matter of fact, some of them have an ethnomedical background and are used by a few ethnic groups in a traditional way for the treatment of gastrointestinal diseases. So, in fact, there are also reports about the antibiotic activity of some of these species. So it was very important for us to determine if some of these species could have an effect against Helicobacter pylori and the pathologies associated to the infection. Well, I wanted to introduce or talk about this bacteria, which is Helicobacter pylori. It's a gram-negative bacteria that colonizes the gastric mucosa, the human gastric mucosa. And to perform this, it has important virulence and colonization factors, such as the enzyme urease, which helps the bacteria to adapt to the harsh acidic environment of the stomach. And besides, the adherence of H. pylori to the gastric epithelium is a primary step for a successful colonization. And this process of binding is mediated by multiple adenosines, which recognize some glycosylated receptors of the host. And with respect to the virulence, such as the vacuolating cytotoxin A, among others, it is very important to consider them as they are closely related with the clinical evolution of the infection. And it is also very important to consider the prevalence of the infection, because the infection by these bacteria represents a very important problem of public health, not only for Mexico, but also for all the world. It is considered that more than 50% of the world's population is infected by H. pylori. However, the prevalence may vary depending on other elements such as the age, gender, the geographical location, or even some socioeconomic factors. Regarding the clinical evolution, it is known that the infection by H. pylori is the main etiological factor of gastrointestinal diseases such as acute and chronic gastritis. Fortunately, in 80% of the cases, the infection may remain asymptomatic, but in the worst cases, it may cause peptic ulcers that are located at the bowel or at the stomach. And also in the worst case, it may become a type of lymphoma associated to the gastric mucosa, but also is associated to the development of gastric cancer. Even though there are therapies for the treatment of peptic ulcers or to eradicate Helicobacter pylori, unfortunately, the treatment fails in about 20% of the cases, and there are also several problems with the current therapies. For example, they are very complex and expensive because it is needed the administration of at least 2 or 3 antibiotics plus a proton pump inhibitor. So this has provoked the appearance of resistant strains. Also, there are several side and adverse effects, so the patients do not complete the treatments, and this generates high rates of recurrences. And there is not an available vaccine yet. So with the aim of determining in vitro the anti-H. pylori potential, we selected 3 representative species of quelites, which are commonly known as alache, chepil, and chaya. So first of all, we tested for their anti-H. pylori activity, and also it was evaluated their potential of having an effect upon 2 of the most important factors of colonization, which is the adherence of the bacteria to a human gastric adenocarcinoma cell line, and also the effect on the enzyme urease of the bacteria. And considering the pathologies associated to the infection, we also evaluated the gastroprotective effect of a mucilage that can be obtained from the aqueous extract of alache and also chepil. So for our purpose, the 3 species of quelites were collected from 3 states of the country, which were the state of Mexico, in the case of Anoda cristata, also commonly known as alache, Nidoscolus aconitifolius, which is commonly known as chaya. It was collected from Yucatan, and Crotalaria pumila, commonly known as chepil, it was collected from Oaxaca. So first of all, the leaves of the plants were dried and pulverized, and with this material, it was obtained an aqueous and a dichloromethane methanol extract. And from now on, I'll be referring to that as DM extract. And the solvents were evaporated to dryness, and the 2 extracts were obtained from the 3 species of quelites were used to test in vitro the anti-H. pylori activity by using the broth dilution method, and it was determined the minimum inhibitory concentration, which is the lowest concentration of the extract that completely inhibited the bacterial growth. And And the other assay that we performed, we tested the effect of the extracts on the adhesion of H. pylori. We used the fluorometric method. So the bacteria H. pylori were labeled with FITC, and it was measured the quantity of H. pylori bound to the AGS cell line in the presence or absence of the different concentrations of the extracts. And also, we evaluated the potential of the extract to inhibit the urease activity. And first, we obtained the enzyme from H. pylori, and then we used the colorimetric method, which led us to measure the ammonia released by the effect of the urease, again, in the presence or absence of different concentrations of the extract. And also, we were able to obtain a mucilage from Alache, Anoda cristata, and Chepil, Crotalaria pumila. In this case, we used the aqueous extract, and from each one of the plants, the mucilage was precipitated with ethanol and finally dried in an oven. And both mucilages were used to test the anti-H. pylori activity, but particularly the mucilage obtained from Alache was used to test the gastroprotective effect in a murine model, which is based in the oral administration of ethanol to induce gastric ulcers. And the mucilage was administered before the induction of the gastric ulcers. So our results, here we have the results obtained with respect to the effect of the extracts and the mucilages of the quelites. It turned out that the DM extracts of the three species of the quelites inhibited better the growth of H. pylori compared with metronidazole, which is one of the antibiotics used for the eradication of the bacteria. And the most effective was the DM extract from chaya, which had the best minimum inhibitory concentration, then followed by the DM extract of Chepil and chaya. And unfortunately, none of the aqueous extracts or the mucilages had activity against H. pylori. Regarding the antiadherent activity, it turned out that all the extracts of the three quelites exhibited an inhibition of the adherence of H. pylori to the AGS cell line. But in this case, the DM extracts from Alache and Chepil showed the best inhibitory effect compared with the controls. So these results are very important for us because one way of avoiding the colonization of H. pylori could be by affecting the adherence of the bacteria to the gastric cells. And considering that until now there is no available vaccine, it could be possible that these quelites may interfere in the initial steps of the colonization. And in the last column, it is shown that none of the tested extracts had an inhibitory effect upon the urease of the bacteria, which means that this enzyme is not a target for the anti-H. pylori activity. Then we made a variation in the methods, taking into account that these plants are usually used as food and they are prepared in different ways. What these three quelites have in common is that for the usage as food or even as beverages, they are submitted to a thermal treatment. For example, for the preparation of the broth with Alache, this quelite is boiled in water for at least 30 minutes. So for our experiments, we submitted the three species of quelites to a thermal treatment, just in the same way as they are cooked or prepared. Then they were dried and grinded into a fine powder, but they were not soluble in the conditions of the experiment. So again, from these three quelites, we obtained their DM extracts and they were tested for their anti-H. pylori activity. And to our surprise, the three quelites exhibited a very good antibacterial activity, being the best DM extract from Alache. And When we compare the anti-H. pylori effect of the DM extract from the plant and with the DM extract obtained after the thermal treatment, it turned out that the thermal treatment improved by much the minimum inhibitory concentration. So this was a very, very interesting result for us. And finally, well, considering our first results with respect to the mucilage from Alache, we used it to test its potential gastroprotective effect in a murine model where the ulceration is induced by the administration of ethanol. For these experiments, the mucilage was administered 30 minutes before the induction of the ulcers with ethanol. We made a microscopic and an histological evaluation of the ulcers, and the analysis showed that the administration of the mucilage protects the gastric mucosa mainly with the dose of 500 milligrams per kilogram of weight. But even with the dose of 300 milligrams per kilogram, it can be observed in the histological images that the mucilage avoids, very importantly, the damage to the gastric mucosa. And with the doses of 400 and 500, it can be seen that the structure of the epithelium is better preserved and there is less inflammatory infiltration. And then in this graph, we can observe that all the doses with mucilage exert a gastroprotective effect, but with a dose of 500 milligrams, we can attain a 56% of gastroprotection, which is as good as a positive control with Carbonex alone. So our conclusions are that the DM extracts of the 3 species of quelites exhibited the best antibiotic activity. The outputs on the DM extracts exert only a mild anti-adherent effect of H. pylori to AGS cells, which was from 30 to 50% of anti-adherent activity. And none of the extracts exhibited an inhibitory effect on the H. pylori urease activity. And very interesting is that the thermal treatment of the plants, like the way they are used as food, improved very much the anti-H. pylori activity. And the mucilage of Alache has a potential gastroprotective activity. And well, finally, I just want to thank Dr. Irma Romero, who is the head of the laboratory, Dr. Amanda Galvez, and also, of course, the students that worked in this project, and the grants from the university. Thank you so much for your attention. If you have any question, I'll be happy to answer it.